Good morning, students. We will continue carbohydrate metabolism, competency number 4.2, and today's topic of discussion is disorders of lipid transport and storage like fatty liver. So today we are studying a topic called as fatty liver. So first, what is fatty liver? Excessive deposition of trialkyl glycerol in the liver is called as fatty liver. Now, what is the mechanism of deposition? Why fatty acid triglycerides are deposited in the liver? It is due to two reasons. First, due to increased synthesis of triglyceride in the liver, and second, due to decreased excretion of triglyceride from the liver to the peripheral tissue. The main cause here is due to decreased synthesis of VLDL. In the last lecture, I have told you that triglycerides are synthesized by the liver and transported to the peripheral tissue by incorporating these triglycerides into the lipoprotein called as VLDL. If the synthesis of VLDL is hampered due to any reason, then the transport of triglyceride does not occur, and the triglycerides which are synthesized by the liver, they remain in the liver itself, or they get deposited in the liver, leading to a disease called as fatty liver. Now these are the causes of fatty liver. First cause is excessive intake of glucose. If you take large amount of carbohydrate, then glucose is formed after digestion of carbohydrates. This glucose through the glycolysis forms glycerol 3-phosphate, and it also forms acetyl-CoA, which is a substrate for de-novo synthesis of fatty acid. So from this glycerol 3-phosphate and fatty acid, they get combined to form triglyceride. Similarly, excessive intake of lipids also helps in lipogenesis by making available the substrate like glycerol and fatty acids. Next causes are diabetes mellitus and starvation. In diabetes mellitus and starvation, there is decreased insulin level, or you can say glucagon to insulin ratio is high. In this case, just we have studied in the last lecture, hormone sensitive lipase is activated. This hormone sensitive lipase acts on the triglycerides present in the peripheral tissue, causing lipolysis of these triglycerides. After breakdown, these fatty acids and glycerol is transported to the liver. In the liver is not under the control of these hormones, insulin and glucagon. So when in the liver, when glycerol and fatty acid is available in large quantity, then esterification occurs and triglycerides are synthesized. So in this way, more triglycerides are synthesized in the liver in diabetes mellitus and starvation. Next cause is deficiency of carnitine. So what is carnitine? Carnitine is a transporter that we have studied during beta oxidation of fatty acid. Here this carnitine transport the fatty acid from cytoplasma to mitochondria for beta oxidation, because in mitochondria the beta oxidation takes place. So if this carnitine is deficient, then the fatty acids which are coming from the adipose tissue by lipolysis enters into the cytoplasma of the liver, but they will not able to enter into the mitochondria for beta oxidation. Therefore these fatty acids get accumulated. As the substrate is accumulated in the cytoplasma, these fatty acids are esterified to form triglyceride. So in this way there is increased synthesis of triglyceride. Excessive intake of alcohol, next cause, also causes fatty liver. The mechanism is, if alcohol intake is more, the formation of acetyl-CoA is more. Now this alcohol is metabolized through formation of acetaldehyde, which is converted into acetic acid, and that acetic acid is then converted into acetyl-CoA. So when acetyl-CoA is formed, then that acetyl-CoA is a substrate for fatty acid synthesis, de novo synthesis of fatty acid. And when fatty acid is available, in the liver, then they are converted into triglycerides. So these are the 6 causes of increased synthesis of fatty acid. Now we will see the other mechanism of fatty liver, that is decreased transport of fatty acid due to decreased VLDL synthesis. First is deficiency of polyunsaturated fatty acid. Now how this polyunsaturated fatty acid deficiency leads to fatty liver? These polyunsaturated fatty acids are present in the phospholipids. They are required for synthesis of phospholipid, mainly lecithin. And this lecithin phospholipid is important for formation of covering of a lipoprotein. Here I have told you that this is the structure of lipoprotein. In the core, hydrophobic lipids are present. Covering is formed by phospholipids. Here mainly lecithin. And in the lecithin, polyunsaturated fatty acid is present. So due to deficiency of polyunsaturated fatty acid, phospholipid synthesis is get hampered, and therefore lipoprotein VLDL synthesis is hampered. And when there is no VLDL, then there is no transport of triglyceride. Similarly, deficiency of choline, betaine and methionine. Choline is the nitrogen base present in lecithin, which is required for phospholipid synthesis. So choline, betaine and methionine deficiency also hampers the synthesis of VLDL. Protein energy malnutrition also leads to decreased synthesis of VLDL. How? Because of protein deficiency, the coverings of lipoproteins are formed in small quantity. Coverings like ApoA, ApoB, ApoC, ApoE. So these are the coverings of the VLDL. They are not synthesized in sufficient quantity. Hence, the VLDL synthesis get decreased. Now, next cause of decreased VLDL synthesis is liver injury or any liver disease like infections, hepatitis, or liver injury due to toxins and poisons like carbon tetrachloride, arsenic, lead, or some drugs like puromycin which inhibits protein synthesis. So all these causes leads to decreased VLDL synthesis. Decreased VLDL synthesis means decreased transport of triglyceride. If the transport does not occur, then triglyceride start accumulating in the liver cells, leading to fatty liver. Now in short, we will see the pathogenesis of fatty liver. When there is excessive triglycerides available in the liver, then they get deposited in the liver cell. Slowly they displace the nucleus from the central position, and a time will come at which they completely exclude the nucleus out of cell, and complete cell is occupied by triglyceride. When the cell is excluded out of cell, the cell dies. Whenever there is cell death, then that dead portion is covered by collagen. So living cells, first they die because of excessive deposition of triglycerides, and then they are covered by collagen tissue. That part of liver is called as cirrhosis of liver. Wherever the living tissue is replaced by collagen tissue, so it is called as liver cirrhosis. So fatty liver may lead to liver cirrhosis if not treated. Now lipotropic factors. So these are the factors which prevent fatty liver. Lipotropic factors are choline, methionine, betaine and inositol. Now how will you manage? We will not go in details, we just discuss about the outline. First we have to treat the underlying cause like diabetes mellitus or any other liver injury. That cause should be treated. Protein energy malnutrition. So first you have to treat the underlying cause of fatty liver. Then you have to give the nutritional therapy. This nutrition therapy is important. Here we have to give carbohydrate rich diet. moderate protein diet. Why proteins are restricted in liver? Because proteins are mainly catabolized in liver. The amino group of amino acid of protein is converted into urea in the liver. So in fatty liver, the liver function is compromised, so urea cycle is also compromised. Therefore amino group is not efficiently converted into urea, and if you take more protein, means more amino groups after catabolism, and that amino group may get converted into ammonia, which is toxic. Therefore we have to avoid a protein, means not avoid, we have to decrease the protein intake, and the proteins preferred in fatty liver is fish proteins. At the same time, the fat should be avoided. Fat should be avoided, and fat should be taken in very small quantity. Saturated fats are completely stopped. The sources of fatty acids are animal fat and coconut oil. They should be completely stopped. So in nutritional therapy, you have to remember that carbohydrate rich, moderate protein and very low fat diet should be given to the patients of fatty liver. So many times a case is asked on fatty liver. So this is one of the example. A 50-year-old male with history of chronic alcoholism had fatty liver. So questions are, what is fatty liver? Excessive deposition of triglycerides in the liver is called as fatty liver. What are the causes of fatty liver? Next question. So these are the causes of fatty liver. Next question is, what are lipotropic factors? Name any two. This is the answer. Lipotropic factors are the substances which prevent fatty liver, and these are the examples of lipotropic factor. And last question is, what is the nutritional theory of fatty liver? Here, carbohydrate rich, moderate protein and low fat diet should be taken, and saturated fatty acid is completely avoided by stopping the intake of animal fat and coconut oil. So this is about the fatty liver. Thank you very much.